How plants sense drought. The crowded truth behind osmotic stress: plants are the ultimate survivors. They can't run away from drought, salt, or freezing temperatures, yet they've evolved clever ways to sense and adapt to these challenges. But how exactly do they do it? A groundbreaking study published in Science on November 1, 2024, by Zhenyu Wang and colleagues from the Southern University of Science and Technology, SUSTech, in Shenzhen, China, reveals a fascinating new mechanism. Plants use a protein called DCP5 to sense changes in water availability through a process called molecular crowding. Plants are constantly exposed to osmotic stress, changes in water availability caused by drought, salinity, or extreme temperatures. When soil dries up or salt levels rise, water rushes out of plant cells, causing them to shrink. This shrinkage disrupts cellular functions, making it harder for plants to grow and survive. While scientists have long known that plants detect osmotic stress through changes in membrane tension, this new study uncovers an entirely different sensing mechanism happening inside the cell. The researchers focused on a protein called decapping-5, DCP5, which is found in the model plant Arabidopsis thaliana. Under normal conditions, DCP5 floats freely in the cell. But when water levels drop, the cell shrinks, and its interior becomes more crowded with molecules. This molecular crowding triggers DCP5 to undergo a dramatic transformation. It forms tiny droplets, or condensates, through a process called phase separation. These condensates, dubbed DCP5-enriched osmotic stress granules, DOSCs, act like emergency control centers. They sequester key molecules, including messenger RNAs, mRNAs, and regulatory proteins, effectively reprogramming the cell's activities to help it survive the stress. For example, DOSCs shut down growth-promoting genes and activate stress response genes, allowing the plant to conserve resources and adapt to harsh conditions. The team discovered that DCP5's ability to sense crowding relies on a unique region of the protein called the intramolecular crowding sensor, ICS. This region is rich in hydrophobic, water-repelling, amino acids, particularly leucine, which help DCP5 clump together when the cell gets crowded. Interestingly, this feature is specific to land plants, suggesting it evolved as a survival strategy for life on dry land. Using advanced imaging techniques, the researchers showed that DCP5 condensates form and dissolve rapidly in response to changes in water availability. This reversibility allows plants to quickly adapt to fluctuating conditions, like a sudden rainstorm after a drought. While the study sheds light on a previously unknown osmosensing mechanism, many questions remain. For instance, how do other proteins and molecules interact with DCP5 condensates? And how do these condensates coordinate with other stress response pathways in the cell? The findings also open exciting opportunities for agricultural innovation. By understanding how DCP5 helps plants cope with osmotic stress, scientists could develop crops that are more resilient to drought and salinity, a pressing need in the face of climate change. The implications of this research extend far beyond basic science. Drought and soil salinity are major threats to global food security, and improving crop resilience could have a profound impact on agriculture. By engineering plants with enhanced DCP5 activity or related proteins, we might one day grow crops that thrive in arid or salty soils, helping to feed a growing population on a warming planet. In the meantime, this study reminds us that even the tiniest molecular changes can have life-saving consequences. Who knew that something as simple as a crowded cell could hold the key to survival?